Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's class we are going to discuss the histology of the muscle. So learning objective for the today's class are to describe the muscle tissue, its classification, the detailed structure of the skeletal muscle histologically, both its physiology and function, and also we will discuss about the neuromuscular junction. So in today's class we are going to discuss each and everything about the skeletal muscle. The muscle is a collection of the muscle fibers supported by the connective tissue. It is mesodermal in origin, and this is the slide of the muscle cross section of the muscle tissue. Different functions of the muscle tissue are the movement of the body, the maintenance of posture, the joint stabilization, and the heat generation. So what are some of the things that we use our muscles for? For example, talking, walking, breathing, smiling, eating, studying, driving, also for digesting, coloring, the riding bikes, the blinking of eyes, the sneezing, raising our hands, swimming. Different functions we do with the help of our muscles. So the special functional characteristics of the muscles are the contractility. The one action is to shorten. The shortening generates the pulling force. Then is the excitability. The nerve fibers causes the electrical impulses to travel. Then extensibility, stretch the contraction of an opposing muscle. Then is the elasticity, recoils passively after being stretched. So there are the different terms we use related to the skeletal muscles and all the muscles types. So one of the term is the sarcolemma. It is actually the plasma membrane. It represents the plasma membrane of the cells, its external lamina and the surrounding reticular lamina. So if you recall from your previous knowledge that the cell membrane also composed of the basal lamina and the reticular lamina. Similarly, sarcolemma over here is also formed of the plasma membrane of the cell, which is called its external lamina, and the surrounding reticular lamina. Both of these form the sarcolemma. Then the cytoplasm of the cells form the sarcoplasm. The smooth endoplasmic reticulum is called the sarcoplasmic reticulum. The mitochondria are called sarcosomes. Then the myofilaments are present. Myofilament interaction is responsible for muscles contraction. Two types of myofilaments are seen. These are the thick and the thin filaments. The muscle tissue is responsible for movements of the body and its parts and for changes in size and shape of its internal organs. So this tissue is characterized by aggregates of these specialized elongated cells arranged in a parallel array that have the primary role of the contraction. So in these two slides, this is the lower magnification. This one is the higher magnification. You can see the smooth the skeletal muscle cells are arranged in a parallel array that is parallel to the longitudinal axis of the muscle, and this is important for their primary role of contraction. Muscle tissue basically composed of the cells called myocytes. Myocytes are elongated in one direction called the muscle fiber. So how we classify the muscle? Basically we classify on the features of the light microscopy as well as the electron microscopy. So these are classified as either striated or non-striated. Again, striated muscles are classified into three types: the skeletal muscle, the visceral striated muscles, and the cardiac muscles. So skeletal muscles are all the muscles of the limbs, the body, the trunk, the back, the abdomen. These all the muscles are skeletal muscle which are under voluntary control. Visceral striated muscles are the skeletal muscles of the tongue, the upper part of the esophagus, the last part of the anal canal. So these muscles are under voluntary control, but they are derived from the pharyngeal arches. That's why given the name the visceral striated muscles. The third type of the muscles are the cardiac muscles, which are the muscles of the heart, and non-striated muscles are the smooth muscles which are involuntary in nature. So types of the muscle tissue according to in the different slides, you can see this one is the skeletal muscle, in which you can see the striations are present. These are the cardiac muscle. In again these muscle you can see the striation, and over here you can understand the intercalated discs which are present. These are the smooth muscle which are non-striated. The types of the muscle tissue. In today's class, we will going to discuss the skeletal muscle in detail. So little bit about all of these muscles. The skeletal are attached to and move the skeleton. These are the skeletal muscles. 40% of the body weight is formed by skeletal muscles. These fibers are present in a multinucleated cell or multinucleated syncytium. Embryologically, they were formed by the muscle, the myoblast, and these myoblasts during development fuses to form the multinucleated syncytium. So cells with the obvious striation, you can see these striations as well over here, and the contractions are voluntary. Cardiac muscle only in the wall of the heart, the cells are striated. You can see the striation in these cells. Contractions are involuntary. Also, you can see the intercalated disc and the branching of the cells as well. And the smooth muscle cells are present in the walls of the hollow organ. They lack the striation and the contractions are involuntary. So skeletal muscles made up of the long cylindrical fibers. They are multinucleated cells. The skeletal muscle is a multinucleated syncytium. In skeletal muscle, each muscle cell, more commonly called muscle fiber, is actually a multinucleated syncytium. So this one is the longitudinal section of the muscle fiber, skeletal muscle fiber, and this one is the cross section of the skeletal muscle fiber. So in this longitudinal section, you can see the nuclei which are present in a form of the row just beneath the sarcolemma. This is the higher magnification of this muscle, in which you can easily see the striations which are present in this. The nuclei are present, and these nuclei are present just beneath the sarcolemma in the space between the two muscle fibrils. You can see the capillary is also present, and also the endomysium is present over here. So these are mature multinucleated cells. The muscle fibers reveals polygonal shape with the diameter of 10 to 100 meters. Connective tissue. Now important is the connective tissue. The skeletal muscle consists of striated muscle held together by the connective tissue. So first of all, the individual muscle fiber which is surrounded by, these are the individual muscle fibers, and these are surrounded by very delicate reticular type of the connective tissue, which is given the name of the endomysium. This endomysium is reticular, and it contains smallest arterioles, venules, and the plexus of the nerves. Then these individual muscle fibers combine to form the muscle fascicles, and these fascicles are in turn covered by the connective tissue, and this connective tissue is called the perimysium. These fascicles, then functional unit of the muscle fibers, they tend to work together to perform the specific functions. So what is the epimysium? That is the sheath of the dense connective tissue that surrounds a collection of the muscle fascicles that constitute the muscle. So whole muscle is covered by epimysium. The epimysium is the dense irregular connective tissue. The perimysium is also thicker in irregular connective tissue, and endomysium is a delicate reticular type of the connective tissue. All of these connective tissue carry blood vessels and nerves with them. In the epimysium, the larger blood vessels are nerves are present. In perimysium, the smaller size, the medium-sized arteries and arterioles and venules along with the nerves are present. In endomysium, the smallest branches, that is capillaries, smallest venules, smallest arterioles will be present. So this is the section in which you can see this is the muscle fiber. This one is the muscle fiber, and it is covered by the endomysium. These muscle fiber combine to form muscle fascicle, and these fascicles are covered by the perimysium. And these different muscle fascicles, they combine to form a muscle, and this muscle is covered by the epimysium. So the different layer, the epimysium, the perimysium, and the endomysium are present. Again, this is the cross section. The histological section, in which you can see individual muscle fibers, the muscle fascicles, and a muscle. The complete muscle is present. So individual muscle fibers are covered by endomysium. The collection forming fascicle is covered by a layer called the perimysium, and the fascicles combine to form a larger muscle which is covered by the epimysium. Again, another diagram you can see, this is the individual muscle fiber, also called the myofibril. These combine to form the muscle fascicles. These are covered by the epimysium, and these muscle fascicles combine to form the muscle itself, and these are covered by the this one is outer is epimysium and inner one is the perimysium. So the skeletal muscle then again classified into different types. The three types of the skeletal muscle, the red, white and intermediate type, can be identified in vivo. The skeletal fibers are characterized by speed of contraction, the enzymatic activity, the metabolic activity. The current classification of skeletal muscle is based on their contractile speed, the enzymatic activity and myosin ATPase reaction and metabolic profile. So on this basis, we classify these muscles into red, white and intermediate type. In a routine histological slides with the hematoxylin and eosine, we are unable to identify these three types of the muscles, the skeletal muscles, but with the help of the special histochemical techniques, we can identify the red, the white and the intermediate muscle type. These histochemical techniques are used for the specific enzymes which are present in muscle tissue. So on the basis of this, also in the naked eye examination can help you to see the red and the white muscles as well. So the main types of the skeletal muscle fibers, the red, the white and the intermediate type. So we will discuss first the red type of the muscle fiber on the basis of the contractile speed, enzymatic activity and fibers myosin ATPase activity. The three types, the one is the type 1 slow oxidative, other type 2 a fast oxidative and glycolytic, and type 3 are fast glycolytic. So what are the type 1 fibers? They are the slow oxidative fibers. Many mitochondria, large number of myoglobin and the cytochrome complexes. The main important feature is this: the large number of the mitochondria, large amount of the myoglobin, which is an oxygen carrying protein in the muscle cell, and cytochrome complexes which are present. So these muscles are slow twitch and fatigue resistant motor unit. This is a very important point because these are the muscles of the back in the humans and helps to maintain the posture. So these are the fatigue resistant and slow twitch muscles. These are type 1 fibers. Then type 2A fibers are the fast oxidative glycolytic fibers, intermediate type. They contain moderate or intermediate amount of the mitochondria, high myoglobin. They are the fast twitch as compared to the slow twitch of the type 1 fibers, and fatigue resistance just like the type 1 fibers. Then the third type of the fibers are the type 2B fibers. These are the fast glycolytic fibers. They are light pink and fresh specimen. They having the less myoglobin and fewer mitochondria. They have a low level of the oxidative enzymes but exhibits high anaerobic enzyme activity and store considerable amount of the glycogen. So these are the different points you should know that in the type 1 fiber they are using the oxidative phosphorylation, while in the case of the type 2B fibers, they have large amount of the glycogen which is stored, and they use the fast glycolytic pathways which are anaerobic in nature to generate the energies. So these fibers are fast twitch as compared to type 1. Fatigue prone as compared to type 1, which are fatigue resistant. These are fatigue prone motor units and they generate high peak muscle tension, the precise and fine movements. So as the movements show, so they are present in the extraocular muscles, the eye muscles, and also the muscles which control movements of the digits in hands. So these are white muscles, type 2B muscles which are present in the eye and the hand, which are fast twitch and slow and fatigue prone muscles. So differences between two types: red containing high control of myoglobin, content of high content of myoglobin and cytochrome. These containing low content of myoglobin and the cytochrome. They having mitochondria many, they having few. They are supplied by the rich blood supply, poor blood supply. They are slow and continuous contraction. They are rapid contraction and fatigable. They are smaller in diameter. These muscles are larger in diameter. Example are postural muscles. Here the examples are the extraocular muscles. These are the histological diagrams of the three types of the muscle: the red muscle fibers containing high mitochondria, high myoglobin, and large amount of the cytochrome. Mixed contain intermediate number, and the white muscle contain low amount of the mitochondria and low cytochrome and myoglobin. So what are the myofibrils? These are the structural and functional subunit of the muscle fiber. The muscle fiber is filled with longitudinally arrayed structural subunits called the myofibrils. So in this you can see these are the muscle fibers, and these are formed by the longitudinally arrayed muscle fibrils. These are called the myofibrils. The myofibrils are composed of bundles of the myofilaments. So these smaller are the myofilaments. This collection of the myofilaments form the myofibrils. These myofilaments are again the thick myofilament, the thin myofilament, and also the elastic, which are the titin, which are present over here as well in case of the skeletal muscles. So structural and functional unit, muscle fiber is filled with longitudinally arrayed structural subunits called myofibrils. The myofibrils composed of the myofilaments, almost same written over here, the thick and the thin, elastic and the titin. So this is the slide showing you the detailed structure of the skeletal muscle fibers. The skeletal muscle fiber basically composed of the actin and myosin. The actin then composed of the actin molecule, small globular actin molecule, and these globular actin molecule arrange themselves to form the F actin or the fibrillar actin. Surrounding the fibrillar actin is the tropomyosin is present, and to attach to the tropomyosin is the troponin complex. This troponin complex then consists of the three subunits: the troponin C, troponin M, and troponin I. Troponin C bind to the calcium, troponin M bind to the myosin, and troponin I is inhibitory in nature. So this is the thin filament. These are having calcium binding sites, the troponin C unit of the troponin molecules, and these are the thick filaments. These thick filaments are formed by the myosin molecules, and these myosin molecules have the long tails and the globular heads which are present. These globular head project from the surface, and these globular head then make the cross bridges with the tropomyosin to help the muscles to contract. So these are the tails of the myosin, and these are the globular head of the myosin. These are the globular the G actin molecule forming the fibrillar actin. This green color is the tropomyosin, and this one is the troponin complex. Again, you can see the cross striation, the structure of the myofibril, and a series of the sarcomere. So the lighter area which is present over here, this lighter area when studied with the polarizing light microscopy, this is the isoreferent to the polarized light, means that it do not change the direction of the polarized light. So this area is called the I band or isotropic. It is around both the Z disc, and it contains the actin myofilaments. The darker area which appear on polarizing light microscopy is anisoreferent. It changes the direction of the polarized light and it appears darker in color. It contains the myosin filaments. So in between the section, in the center of the light area, you can see the Z disc are present. To these Z disc, the actin filaments are attached, and in the center of the myosin or the thick area, you can see a lighter area which is present in the center. This is the area is called the H zone, and in H zone a fine line is present which is called the M line. So in this detailed structure, you can see this one is the Z disc, and to Z disc actin filaments are attached directly, while the myosin filaments are attached to Z disc by means of the titin. Then you can see this area where both the actin and myosin are present. This is the A band or the darker area, and over here in this area, which is again the lighter area, here you are seeing the lighter area, the H zone. In this area only the tail area portion of the myosin molecules are present, and in the center of this the H zone is present. So these are the different sections which are taken from different area. This is the Z disc, and in this Z disc you can see, in addition to the actin filament, the connection filament which are shown in red color are also present, which are the titin which attaches the myosin to the Z disc. Then the I band, again in the I band you can see the actin filaments along with the titin. Then in the M line you can see only the thick filaments which are present in the H zone, in which tail areas are present, and A band where you can see the tail and head both of the actin myosin filaments, also the actin filaments are also present. So events of the muscle contraction, this is very important point in physiology, you must understand. So we do it systematically. First of all, a nerve impulse triggers the release of acetylcholine from the synaptic knob into synaptic cleft. The acetylcholine bind to acetylcholine receptors in motor end plate of neuromuscular junction, initiating the muscle impulse in the sarcolemma and the muscle fibers. So here you can see this is the neuromuscular junction. Here you can see this is the nerve ending, and in this nerve ending, a nerve terminal, many synaptic vesicles are present, and these synaptic vesicles, upon stimulation, they are releasing the acetylcholine. There are many acetylcholine receptors over here in the muscle where this neuromuscular junction is present. These acetylcholine bind to acetylcholine receptors which activate the sarcolemma of the muscle. The next step is, as the muscle impulse spreads quickly from sarcolemma along the T tubules, calcium ions are released from the terminal cisternae into the sarcolemma. So here you can see this is the terminal cisternae of the sarcolemma, and when action potential spread over here, it also spread into deeper layers of all the muscles, and the sarcolemma this T tubule then secrete a large amount of the calcium ions. So next step is third step is the calcium ion binds to the troponin. So troponin molecule have calcium ion binding sites. Then calcium ion binds to these troponin. Troponin changes its character, it changes its shape, it moves the myosin on the actin to expose the active sites on the actin molecule. So because of the changes, when calcium ion binds to troponin, and troponin changes the position of the tropomyosin, as a result of this, the active sites which were blocked by the tropomyosin, now they are exposed, and because of this exposure, the myosin now making the cross bridges with the active sites on the actin molecules. When impulse stops, calcium ions are actively transported into sarcoplasmic reticulum. Tropomyosin recovers the active sites, and filaments passively slide back to their relaxed state. Also, very important point you must remember. These actin molecules are having the adenosine triphosphate is broken in the form of the ADP and phosphate enzyme, which is high energy releasing product, and this is attached to the actin. Upon stimulation, upon attachment of the calcium ion to the troponin C complex of the actin, this they releases the adenosine diphosphate and phosphate ion. As a result of this, there is the conformational changes and active sites are released, and the heads of the myosin attaches to active site on the fibrillar protein on G proteins areas. When they attaches to these areas, again new ATP is available, and when this new ATP binds to the troponin C part of the troponin molecule, again the conformational changes takes place and the recovery strokes takes place. As a result of this, these cross bridges are broken, and the myosin molecules move further ahead to contract the length of the sarcomere. So this is the attachment of the energy is provided by the ATPase enzyme which is present in the troponin C molecules, and this ATP is cleaves the ATP into ADP and the phosphatide and release high energy, and this energy is required for contraction of the troponin molecule to the myosin molecules. So myosin head pivot, moving thin filaments towards the sarcomere center. ATP binds the myosin heads and is broken down into ADP and phosphates. Myosin heads detach from the thin filaments and return to the pivot position. The repetitive cycle of attach, pivot, detach, return, slides, thicken, thin filament past one another. So the sarcomere shortens and muscle contract. The cycle continues as long as the calcium ions remain bound to the troponin to keep active sites open. Again, another very important point you must remember that the troponin molecule, the actin molecule and the myosin, they do not shorten. They actually slide past along each other to decrease the length of the sarcomere. So sarcomere length, which is normally 2 to 3 micrometer, it decreases in contracted states to 1 micrometer. They slide past each other, but actually their own length do not decreases. This is very important point to understand. Again, another slide showing you the this is the H&E stain section, showing the cross striation, the position of the nuclei which is present just beneath the lemma, the plasma lemma. Here you can see the detailed structure. These are the smaller myofilaments which are present, and the surrounding these myofilaments in the plasma lemma, large amount of the mitochondria. The rough endoplasmic reticulum is present. This is a small myofibril which is present, and this is the detailed structure of the myofibril. This is the Z disc. This in the center is the actin filaments are present, which is lighter area called A band. This is the darker area called I band. In the center you can see the H zone, and in the center of the H zone you can see the M line which is present. Again, this striated muscle, here you can see the C disc which is present, the actin filament, the myosin filaments, the tail area of the myosin filament which is forming the H band, and in the center you can see the M line. So the sliding filament model, here you can see the relaxed sarcomere, the partly contracted and the fully contracted. In relaxed you can see the full length of the sarcomere. Here you can see the sliding of the actin myosin filaments along each other, and in contracted you can see the actin filaments moving towards the H zone in the middle of the fiber, making the cross bridges with the myosin. So this is another picture showing you the thick and thin filaments. So these are thin filaments, these are thick filaments, and this is the contracted state. The thin filaments diagrammatically to show the fibrillar protein, the myosin head making cross bridges with the filaments. And these are the electron microscopic picture in which you can see the thick are the myosin filaments and the thin filaments which are present, and the cross bridges between the two you can see easily. An electron microscopic picture of the myofibrils. This one is unlabeled. Here you can see labeling. This one is the first. This is the Z disc, then the A band, I band, A band, H zone, and M line. So sarcoplasmic reticulum, we are repeatedly talking about the sarcoplasmic reticulum, we are repeatedly talking about the calcium ions, we are repeatedly talking about the action potential which is generated, which spreads along the all muscle fibers very rapidly. So what is the mechanism which helps this action potential to spread rapidly to release calcium levels to all the levels of the muscle? So this is the sarcoplasmic reticulum, which is very important. It stores a large amount of the calcium ion in itself. So how it is organized? The extension of the sarcolemma called the T-tubules or transverse tubule extend between the parallel cisternae of the sarcoplasmic reticulum that encircle the individual myofilaments, allow very rapid transmission of the surface action potential to the interior of the cell, releasing calcium ion from sarcoplasmic reticulum and allowing the sarcomeres of the myofilaments to all contract as essentially the same. The T-tubules here you can see they are surrounding the myofibrils and they are forming the network. So the one of the T-tubule, one of the sarcoplasmic reticulum surrounding the myosin, the darker area and the lighter area, and this is the area where the dark and light area are present. And here you can see the dilated part of the sarcoplasmic reticulum which is forming the cisternae. Also very important is the plasma lemma, which forms a transverse tubule or T-tubule, which is present over here. It is present, it goes deeper into the muscle myofibrils at all the levels, and the collection of these two cisternae, and in the center the T-tubules, they are forming the triad. So action potential is when it is generated, it rapidly reaches all the level of the myofibril with the help of the T-tubules, and the because of the activation, the sarcoplasmic reticulum large, which is storing the calcium ion, it releases a large amount of the calcium ion. This calcium ion then stimulates the action potential. This stimulates the contraction of the thin filaments and the triggering the sliding filament mechanism of the contraction. So T-tubules are continuous with the sarcolemma. Therefore, whole muscle, the T part as well, contract simultaneously. This is a very important point, because all of the muscle has to contract simultaneously and has to generate a strong force. That is why it is required that the action potential to reach at all levels simultaneously, and it is with the help of the T-tubules which are present in this yellow color. You can see these T-tubules. It is present in the deeper part, and it is responsible for the spread of action potential to all levels of the muscle at the same time. So again you can see very important here, these are the sarcoplasmic reticulum, these are the network of the sarcoplasmic reticulum, and at the junction of the lighter and the darker band you can see a structure called the triad. Here the sarcoplasmic reticulum dilated to form the terminal cisternae. On both sides you can see terminal cisternae, and in the center you can appreciate the transverse or the T-tubule. These T-tubules are derived from the plasma lemma. Next is the neuromuscular junction, again very important. The motor neurons innervate the muscle cell fibers. Here you can see these are the motor neurons which are starting at the higher center in the cerebral cortex, passing to the spinal cord, to the motor nerve, to the skeletal muscle. At the level of the skeletal muscles, these axons divide into many terminal branches which surround the skeletal muscle. So this is the motor end plate where it meets the skeletal muscle, and here it stores large number of the vesicles, neurotransmitter containing vesicles. The neurotransmitter at this site is the acetylcholine. Upon stimulation, these acetylcholine vesicles bind to the cell membrane, and by process of the exocytosis, they release the acetylcholine. There are large number of the folding of the plasma membrane in relation to the motor end plate, and it contains larger number of the acetylcholine receptors. So these acetylcholine receptors bind to acetylcholine. Acetylcholine binds to acetylcholine receptor and stimulate the plasma membrane. This because of the stimulation of the plasma membrane, nerve signals are generated, and these nerve signals releases the sodium ion throughout the plasma lemma. This sodium ion then stimulate the terminal cisternae of the endoplasmic reticulum at all the levels of the cell of the skeletal muscles, and as a result of this stimulation, the sarcoplasmic reticulum releases releases the larger amounts of the calcium ion into the plasma, into the sarcoplasm, as a result of which the excitation, contraction, coupling take place. So motor unit is the motor neuron and all the muscle fiber it innervates. This all area is called motor unit. Average is about 150, but range is 4 to several hundred muscle fibers in a motor unit. The finer the movement, the fewer muscle fibers and motor unit. So this you must understand, where very fine and very precise movements are required, like extraocular muscles, like the muscles of the digits. The fewer muscle fibers are supplied by the fewer units, while in cases of the skeletal muscle, the nerve terminal divides into many branches, and a single axonal branches can supply large number of the muscle fibers. So for precise movement, every muscle or every one or two muscle will receive a single axon. The fibers are spread throughout the muscle, so stimulation of the single motor unit causes a weak contraction of the entire muscle. So as a result, all of the muscle are stimulated at the same time by a large number of the motor axons which supply the muscles. This is detailed structure. Again, the larger section of the neuromuscular transmission. This is the nerve terminal containing acetylcholine vesicles. Acetylcholine is released by the exocytosis. Large number of the receptors which are present at the junction at the motor end plate, and this acetylcholine attaches to it, at a result of the which action potential is generated. This action potential then stimulates, passes through the tubule to the sarcoplasmic reticulum, and then sarcoplasmic reticulum release calcium ions. So these are the histological slides. In the longitudinal section, you can see the muscle fibrils or myofibrils are arranged in parallel to the long axis of the muscles. Here you can see the cross striation. Again, very important point to understand, to identify the slide histologically, is that all the muscle fibers having the same diameter. And then another important point is the position of the nuclei, which is present at the periphery just beneath the sarcolemma. So when you see the cross section at these level, you can see almost all the muscle fibers having the same diameter and their nuclei are present at the periphery. This is the detailed section of one of these, and here you can see this dotted appearance. This dotted appearance actually shows you the cut ends of the actin and myosin filaments which are present. This is the electron microscopic picture of the skeletal muscles. Here you can see the Z discs which are present, the lighter area and the dark area. Here you can see the mitochondria as well. So here I complete my today's class. Thank you very much for your attention. The references for today's class are Basic Histology by Laiko Siddiq, the Histology text and atlas by Jan Quera, and Histology text and atlas by H. Ross. Thank you very much for your attention, and Allah Hafiz till the next class.